Susan and Sharon are identical twins who have the same DNA, genetic information stored in the cells in a certain order or sequence that serves as a code to tell their bodies how to function. DNA determines what the genes do, the chemicals they produce, and what health risks they're predisposed to throughout their lives. Even though Susan and Sharon have the same genetic information, they're not alike in their personalities and behaviors, or in their health as they age. Susan gains weight and has trouble losing it, while Sharon maintains a healthy weight throughout her life. Susan develops cardiovascular disease, while Sharon doesn't. If they have the same genes, how can that happen? Clearly, DNA is not our destiny. New evidence shows that we can change the effect of our genetic makeup on our health. For example, when pregnant, women's experiences, including behavior, diet, and environment, can affect the gene expression of their child without changing the child's DNA sequence. The influence of external factors on our genes is called epigenetics. In addition to influencing which of our genes express themselves and how, epigenetic mechanisms may play an important role in the development, diagnosis, and treatment of diseases. By understanding and influencing epigenetic mechanisms, we can optimally experience the 4 crucial components of our gene expressions for a healthy life. Potential: our gene risk factors and influences. Capacity: external effects on metabolic processes and our health status. Balance: through critical nutrition management and behavior change, we can maintain optimal methylation for our genes to be most favorable for health. Expression: our optimally managed health status. There are 2 important epigenetic mechanisms: DNA methylation and histone modification. The first is histone modification. In all of our cells, our DNA is wrapped around proteins called histones. When the DNA is wrapped tightly around the histones, the genes are unreadable and they are not expressed. When the DNA strand is loosened around the histones, genes are accessible and may be read and expressed. In this way, modification of histone proteins can turn a gene's expression on or off. DNA methylation occurs when chemical switches called methyl groups are added to or removed from regions of the DNA. Methyl groups affect the expression of the genes in that region of DNA by turning them on or off. These 2 epigenetic mechanisms can explain why Sharon and Susan, though they have identical genes, have different health outcomes. Their different environmental exposures or their health behaviors may have led to different epigenetic influences, and therefore to their different traits and health outcomes. Epigenetic mechanisms like histone modification and DNA methylation are complex. The DNA methylation process involves several metabolic gears that produce the necessary methyl groups for the methylation process. Each component or gear in the methylation cycle is important for the process to continue, and several nutrients are required, including vitamin B12, vitamin B6, and folic acid. When there are non-optimal levels of folic acid in the body, the gears of the methylation cycle don't work properly. When they move too slowly, the methylation process is disrupted. Different diseases may develop, including cancer, cardiovascular disease, Alzheimer's disease, and diabetes. One example of when the gears of the methylation cycle could slow down is when a person has a variation of the gene MTHFR. A variation, or polymorphism of this gene, causes metabolism of folate to be changed. Variations in MTHFR are common in the population and may affect up to 40% of us. People with MTHFR polymorphisms have increased risk for several diseases, including cardiovascular disease and some cancers. Outcomes that can be influenced by genetic polymorphisms especially affect people who have 2 copies of the polymorphism in their genetic code, also known as homozygous expression. Having 2 copies of the genetic polymorphism means that both parents passed on the gene polymorphism to your genes. Being heterozygous, having just 1 copy of the polymorphism, means the risk of disrupted methylation is reduced but still present. Identifying genetic variations and our methylation capacity may be done by simple assays of blood. Blood is easy to access and can reflect metabolic and inflammatory pathways and outcomes in our cells, including levels of important blood chemicals like homocysteine, S-adenosylmethionine, and S-adenosylhomocysteine. Identifying methylation capacity and balance in blood chemicals with these assays allows healthcare providers to identify high-risk patients like Susan, who may benefit from health interventions. Nutritional interventions and therapies like adding folate to the diet target and may improve the methylation process. Unlike mutations and other genetic abnormalities, epigenetic modifications for our gene expression may be changed by changing our behavior. The study of epigenetic markers and mechanisms is emerging as one of the most promising strategies for identifying and treating patients who have or may develop complex diseases. When genetic testing and related therapies are implemented, we will be able to improve public health.